So, hello everyone. This is Dr. Manar Hajir, a histopathologist from Jordan University Hospital. Today's lecture will be about apoptosis and autophagy. But apoptosis is defined as a programmed cell death. We have two types of cell death. One is called necrosis and the other one is called apoptosis. And they are different from each other, in which apoptosis is considered a programmed one, genetically determined process of self destruction of the cell, and this pathway is highly controlled, in which the cell will activate enzymes that degrades its own nuclear DNA and nuclear and cytoplasmic proteins. Now, the dead cells and their fragments are cleared with little leakage or no leakage of cellular content to the outside of the cell, so there will be no inflammatory reaction, and this is one difference from necrosis pattern of cell death. So this process of apoptosis is a highly controlled, highly predetermined, genetically determined process. Sometimes we call it also cell suicide or the clean cell death. As you can see here in this picture, the first column or the column to the left represents the pattern of cell death by necrosis, and to the right we have the cell death by apoptosis. And as you can see here, we have too many differences between these two pathways of cell death. Necrotic pathway is discussed in a separate lecture, and here you can see one of the main differences between apoptosis and necrosis is that in apoptosis the cell underwent shrinkage instead of swelling, and we start to have many blips of the cell membrane. Uh, the nuclear material starts to condensate. The chromatin becomes darker under the light microscope, and then it starts to fragment, as we call it, karyophytes. And as you can see here, also the cell membrane stays intact, and the cell starts to fall off. That's why it is called apoptosis. Apoptosis means cell falling off. It's like عنقود العنب, and every part of the cell membrane encloses some organelles and some cytoplasmic proteins, and it separates from the main cell. Then these fragments or apoptotic bodies, we call them, they give some signals to the phagocytes that will come to engulf them and get rid of them without eliciting an inflammatory reaction. So again, here we have the normal cell. The cell undergoing apoptosis first shrinks in size, decreases in size, and it starts to fall off in the form of apoptotic bodies, which are membrane-bound vesicles or fragments of the cell. The nucleus, as you can see here, undergoes also certain changes in the form of shrinkage, then fragmentation of the nucleus, and as you can see here, every apoptotic body contains parts of the organelles, parts of the cytoplasmic proteins, parts of the nucleus, fragmented nucleus or DNA material, and the phagocytes will sense the presence of these vacuoles or apoptotic bodies and will come to eat them, let's say, or clean the area without eliciting again an intense inflammatory reaction. That's why we call apoptosis the clean cell death. In this table, you have to know the main differences between necrosis and apoptosis. In terms of cell size, the nucleus, the cell is in necrosis is swollen, and in apoptosis it is shrinked. In terms of nuclear changes, in necrosis we have pycnosis, karyophytes, then karyolysis. In apoptosis, we have we can say that there is pycnosis, then karyophytes, then these karyorectic debris of the nucleus will start to separate in the apoptotic bodies, and these apoptotic bodies will then be cleared by the phagocytes. Now, the plasma membrane, this is an important difference. It is disrupted, it is damaged in necrosis. That's why the enzymatic content of the cell will leak to the outside and sometimes will gain access to the bloodstream. While in apoptosis, the membrane is intact and the cellular contents, cellular proteins, cellular enzymes are retained within the cell. So there will be an adjacent inflammatory reaction in cases of necrosis because of this leakage of content, while in apoptosis the area is clean. The number of cells dying by apoptosis usually not too many cells, not like in necrosis, and we have no adjacent inflammation in apoptosis. Now, again, also necrosis, it's almost always pathologic, while in apoptosis, you will find during this lecture that we have physiologic causes and pathologic causes of apoptosis. Sometimes apoptosis and necrosis, they can coexist, and apoptosis induced by some pathologic stimuli may progress later on to massive necrosis, like in cases of ischemia, for example. In this picture here, it is an H&E stain section under the light microscope. This is what we used to see under in our pathology lab. Here you can see these cells, as you can see here, it is shrunken. There is an empty lacunae around the cell here, and the nuclear material is fragmented, it is condensed, it is very dark in color, and there are a few cells that die by this pattern. As you can see here also, there are fragments of the DNA material, and you don't see an inflammatory reaction. Now, causes of apoptosis, we have physiologic, normal scenarios in which apoptotic cell death takes place, and we have pathologic conditions. Examples on the physiologic forms of apoptosis, one of them is the during embryogenesis, during the embryo development in the uterus. All the time during embryogenesis, new cells appear and old cells die, and this death is usually occurs by means of apoptosis. Another example is the involution of tissues upon hormone deprivation, like what we see in the cyclic endometrium at the end of the menstrual cycle. The endometrial glands are lost, and then the shedding of the endometrium will start as menses. This loss and this death is usually mediated by apoptosis. Also, in the lactating breast, after the cessation of lactation, there will be deprivation of the hormone, and the new glands which appeared at the onset of lactation will involute and will die by the process of apoptosis. So this is mediated by the loss of hormonal stimulation. Also, another example is the steady state population in certain organs, like in the gut and the GI tract, gastrointestinal tract, and in the skin. As you know, these organs, they have a rapid turnover, so new cells are made all the time, and the old cells die by apoptosis in a rapid regenerative capacity process. Another example is the end of function or end of life. Sometimes the cells perform their function and there is no need for them anymore, so they should die by apoptosis. وإكزامبل على ذلك هو the neutrophils at the end of the acute inflammatory reaction. These neutrophils have left the blood vascular space into the tissue. When they perform their function, they will die by apoptosis. They will not return back to the blood vessels or to the bloodstream another time. They will perform their function in the inflammatory action and they will die by apoptosis. Also, the death of self-reactive lymphocytes during development or of lymphocytes in the bone marrow or in the lymphoproliferative organs. Sometimes these new lymphocytes could have some activity against the self antigens. So if these lymphocytes stay in the body they will cause autoimmune diseases أمراض المناعة ضد الذات وبالتالي إحنا أي self-reacting lymphocyte بتظهر during the development of or the maturation process of the lymphocytes, this cell should die and the death is mediated by apoptosis in order not to have later on autoimmune diseases. فكل هاي الأمثلة اللي أنا حكيتها هنا they are physiologic causes of apoptosis. Now, the pathologic causes of apoptosis any damaged cell beyond repair بحيث إنه الجسم ما بيقدر يعمل يعمل repair للدمج اللي صار. This cell is targeted to death by apoptosis. Examples on that the DNA damaged cells. DNA damage is usually caused by radiotherapy, chemotherapy, العلاج بالأشعة والعلاج الكيماوي in cancer patients, extremes of temperature sometimes, exposure to the UV light, hypoxia, all of these scenarios can lead to DNA damage. And if this DNA damage is not repaired, the cell will be directed to apoptosis because of DNA damaged cells continues to replicate, we will have cancers at the end. Another pathologic example is the cells that accumulate misfolded proteins, and this will be discussed in the mechanisms of cell death lecture. Any cell that accumulates these misfolded proteins will be directed to cell death by apoptosis. Some infections, especially viral infections like infection by adenovirus, by the HIV virus, AIDS virus, and by the hepatitis viruses اللي بيعملوا التهاب الكبد الوبائي, those viruses they can cause cell death by even by necrosis or by apoptosis. So you will find both of the processes taking place together. So these are the pathologic underlying causes for apoptosis. Now, as you can see in this table, it summarizes the physiologic and the pathologic causes of apoptosis that I have already mentioned. The physiologic during embryogenesis, the turnover of proliferative tissues حكينا منهم, the intestinal epithelium, the skin كمان حكيت لكم, even the lymphocytes in the bone marrow, the thymus, هذولا الأورجانز دائما فيهم proliferation عالي, فبالتالي في cells بتموت وفي cells تظهر بدالها. The cells that die, they die by apoptosis. And this is normal physiologic process. Involution of hormone-dependent tissues like the endometrium at the end of the menstrual cycle, decline of leukocyte numbers at the end of the immune reaction or inflammatory response like the death of neutrophils at the end of the acute inflammation, and elimination of self-reactive lymphocytes. The pathology causes any DNA damaged cell or the cells that accumulate misfolded proteins or cells that are virally infected, those cells will be directed to death by apoptosis, and this is called here pathologic apoptosis. Now, what are the mechanisms of apoptosis? Apoptosis in general leads to activation of enzymes called caspases. But this activation takes place by two pathways. The first pathway is called the mitochondrial or the intrinsic pathway. It's called intrinsic because it starts at the level of the mitochondria inside the cell, and the death receptor pathway. So these two pathways will overlap at the end by activation of the executive caspases, but each pathway has its own distinct properties. The first one is called the mitochondrial or the intrinsic pathway, and it is responsible for apoptosis in most pathophysiologic and pathologic situations. فمعظم حالات الابoptosis اللي بتصير هي by the mitochondrial pathway, and it is regulated by a group of proteins called the BCL2 family of proteins. البروتوتايب بهاي المجموعة أو بهاي الفاميلي هو ال BCL2 اللي بنعتبره إحنا it's an anti-apoptotic protein. Which is encoded by an anti-apoptotic gene. شو يعني anti-apoptotic؟ يعني it prevents apoptosis. This BCL2 family of proteins controls the mitochondrial membrane permeability. ليش؟ لأنه inside the mitochondria we have certain enzymes and certain molecules that if they leak from the mitochondria to the cytoplasm, if the mitochondrial permeability increases, they will cause activation of caspases. And this molecule, the most important molecule of them is the cytochrome C. So the cytochrome C اللي هنا عندكم مبين as a red circle, it is present inside the mitochondria. فهو away, محفوظ away from the cytoplasm. If in any case the mitochondrial permeability increases, the cytochrome C will leak to the cytosol or to the cytoplasm, and it will cause activation of كاسبيزز وبالتالي apoptosis. فاللي مطلوب في النورمال سيناريو في الوضع الطبيعي انه السيتوكروم سي يضل موجود داخل الميتوكوندريا عشان الخلية تضلها viable and alive. This BCL2 family of proteins includes some anti-apoptotic زي ال BCL2 وال BCLXL، وفي عنا pro-apoptotic اللي هم ال BAX and BAC، وهذول موجودين كلهم في الممبرين of the mitochondria. وفي عنا BH3 sensors عبارة عن proteins موجودة في السيتوبلازم. They will sense any change that will lead to apoptosis. So here, as you can see in the viable cell, the survival signals continue to reach to the cell, like growth factors, like hormones, for example, and this will lead to activation of the anti-apoptotic protein. So بتضلها ال BCL2 active. This BCL2 بيعمل inhibitory effect على ال BAX and BAC اللي هو باللون الأخضر موجود بالممbrane of the mitochondria. فبالتالي this will inhibit the leakage of cytochrome C from inside the mitochondria to the cytoplasm, so the cell will stay alive. This is the cell survival signal. Once the cell is exposed to lack of these survival signals، بطل يوصلها growth factors، تعرضت ل radiation for example، صار في عنا DNA damage، صار في عنا misfolded protein accumulation، كل هاي الأمور بتخلي عندي activation للسنسورز اللي هم ال BH3 family of proteins اللي موجودة في السيتوبلازم. These sensors when they are activated they will inhibit the BCL2 اللي هو بيعملوا antagonization to the BCL2 which is the anti-apoptotic، وبيعملوا activation بالعكس لمين؟ للBAX and BAC. Now BAX and BAC لما يتحرروا من ال inhibition of BCL2 they will dimerize اللون الأخضر اللي انتم شايفينه هنا channel لونها أخضر فاتح light green in the membrane of the mitochondria here, and it will the BAX and BAC will dimerize forming a channel through which the cytochrome C will leak to the outside to the cytoplasm and will cause activation of caspases. It will start with caspase 9, then a series of caspase activation until the process of a cleavage of the cellular proteins and the cellular organelles leading to apoptotic cell death. The other pathway which we call it the extrinsic pathway or the death receptor pathway وسميناه بهذا الاسم because it is a receptor that is present in the cell membrane and it has a domain in the cytoplasm which is called the death domain because it interacts with proteins inside the cell that lead to activation of the caspases اللي هم death enzymes. This receptor family we call them tumor necrosis factor receptor family, and the prototypes are type 1 tumor necrosis factor receptor and the FAS receptor. Regarding the FAS receptor, it has a FAS ligand. This FAS ligand is present on the activated T lymphocytes. What happens is when the FAS and the FAS ligand interact with each other like a lock and key phenomena يعني the receptor and the ligand. They will activate the death domain of the FAS receptor and this will activate an cytoplasmic enzyme called the caspase 8 which will activate a series of caspases that will lead at the end to apoptosis. This pathway is used in much limited circumstances, like in elimination of self-reactive lymphocytes and in the killing of target cells by some cytotoxic T lymphocytes. مين هم التارجت cells في هاي الحالة؟ ممكن يكونوا virally infected cells، ممكن يكونوا tumor cells. تتعرف عليهم السيتوتوكسيك تي ليمفوسايت، بصير الانتراكشن بين الفاس والفاس ليجند، وبعدين تصير activation للسايت للكاسبيز، وبعدين بصير في عندي series of interactions in which the cell will degrade its nuclear material and its proteins and it will fall off as apoptotic bodies. So this is here the Fas ligand اللي بيرتبط بالفاس receptor اللي موجود على the surface of the cell that will die أو is directed to death by apoptosis. This is the death domain, the red in color that will activate, if activated, it will activate the caspases, caspase 8 mainly, that will affect or activate a series of caspase enzymes, leading at the end to apoptosis. As you can see, both the mitochondrial pathway, which is called the intrinsic pathway, and the death receptor pathway, which is called the extrinsic pathway, both of them um, uh, will link at the process of executive caspases activation that will lead to breakdown of the cytoskeleton, endonuclease activation and fragmentation of the DNA. At the end, there will be formation of apoptotic bodies. These apoptotic bodies, they have a message, they give a message or receptor or an attractive process to the phagocytes in order to come and engulf them and get rid of them. As I said, mitochondrial pathway is the pathway responsible for most of cases of apoptosis, any growth receptor withdrawal, DNA damage, accumulation of protein, misfolded proteins in the cells. All of these will lead to mitochondrial pathway activation, but the end result of both pathways is the same. Now let's move to another topic, which is autophagy, which is a related somehow to related process to apoptosis and also related process to the adaptive mechanism, which is called atrophy. Now, autophagy, it means self-eating, that the cell eats itself by lysosomal digestion of the cell's own components. And this is considered a survival mechanism in times of nutrient deprivation or in times of starvation of the cell. The cell will recycle its contents in order to provide the nutrients and energy in times of starvation. What happens in autophagy is that membrane-bound vacuoles, they form in the cell, and those vacuoles are derived from the endoplasmic reticulum membranes. These are called autophagic vacuoles. These vacuoles will fuse with the lysosomes to form what is called autophagolysosome. Then the lysosomal enzymes will start the process of digestion and damage to these cell components. The end result will be shrinkage of the cell and atrophy of the cell, and this is considered an adaptive mechanism sometimes. Now, if there is a failure of adaptation, if the starvation continues for a longer period of time, in which the cell cannot adapt anymore, then a trigger for apoptosis will take place. So as you can see here, a membrane-bound vacuole is formed in the cases of a nutrient depletion. This membrane-bound vacuole, the membrane of which is derived from the endoplasmic reticulum membrane, forming the autophagic vacuole, which will combine with the lysosome and form the autophagolysosome. Then the process of damage and digestion will start. And thank you for listening.